So hello and good evening, everyone. So welcome to another live session on Edentu. Now in Edentu, we have been doing a series of sessions on like how to prepare for your for each subject for your NEET MDS preparation. Okay, so we had already done about the preparation strategies for oral radiology, dental materials, orthodontics, and all. If you wish to see those videos, you can see it under the live session on Edentu YouTube channel. Now, today, we will be mainly focusing on the preparation strategies for oral histology. Okay. So, if you are someone who is preparing for your NEET MDS 2025 examination and has not yet started oral histology subject, or you are in the middle of the preparation for your oral histology subject, then I will tell you the general weightage of this oral histology subject. Okay, like what is it? How many questions can be expected from oral histology subject, and what are the type of questions they ask in oral histology subject, and what are the high yield areas in this oral histology subject, and all those things I'll be sharing with you today. Okay, so oral histology, it is a basic subject, it is a fundamental subject, and it is an interesting subject also. As I had already told you, since it's a basic subject, you must be very thorough with all your basic and fundamental subjects, because then only your clinical subjects will be easier for you. Okay. So, oral histology is a scoring subject. It is a easy subject, and the information that you study in oral histology is easily retainable also. Okay, it's not a volatile subject. It is easily retainable subject also. So the usually one-liner factual based questions are usually asked from the oral histology subject, but sometimes you can also expect the image based and the application based questions also from this oral histology subject. Now I will tell you like what are the types of image based and application based questions. You must be wondering, like, this is a basic non-clinical subject, so how can they ask like application based question or the clinical type of questions in this from this subject? Okay, so I'll be telling you that like what are the kind of questions they can ask from this oral histology. I'll be also explaining that to you. So in this session, we'll be mainly discussing about the general weightage. Okay, general weightage of this oral histology subject, like how many questions can be asked from oral histology, and then we will move on to the previous trend. Okay, like how many questions were asked in this or from this oral histology subject from the year 2017 till 2024. Okay, we are also including the recent exam questions, and we'll be seeing like generally the trend of the questions that are asked from this oral histology subject. Okay, then next what we are going to do is we will analyze each and every question that is asked from 2017 till 2024. Okay, so we will be like analyzing each and every question that is asked from 2017 till 2024, and then we will be assigning them to the particular topic from which this question is asked, and so that at the end we will see like from which topic more questions were asked frequently, or from which topic more questions were asked repeatedly. Okay, so why we are doing this is because it is to simplify your learning process. Okay, it is to like effectively use your study time. Okay, now only like 5 to 6 months are remaining for the NEET MDS 2025 examination, and it is not practically possible for you to like read the theory of like all the 19 subjects. Okay, that's not a practical solution. So what you can do is like you can focus on the high yield topics or the high yield areas from where more questions are frequently asked. Okay, so if you focus on such areas, it will be easier for you to study, and you can manage your study time more effectively also. Okay, so that's what we are trying to help you with here. We will analyze the high yield areas and the high yield topics for you, and we will tell you that these are the high yield topics after analyzing all the questions in front of you from 2017 till 2024, and we will tell you, okay, these are the topics that you should be focusing more on. Okay, And we will also see how many like image-based and clinical-based or application-based questions were asked from the subject of oral histology. Okay, so that's the main thing that we are going to do today. So let's begin, doctors. So, like I had told you, oral histology is a scoring subject. If you are a little bit careful and if you do it properly, then you can really score full marks from the subject of oral histology. Okay. So, according to the National Board website, you can expect a total of like 7 questions from oral histology. So oral histology together with dental anatomy, you can expect a total of like 14 to 15 questions. From oral histology alone, you can expect a total of like 7 to 8 questions. Okay. If the number of oral histology questions are less, then the dental anatomy questions will be more. Okay. And vice versa. If the number of dental anatomy questions are more, then the number of the oral histology questions will be less. Okay, that will be generally seen. Okay, now let's look at the previous trend of these questions from the oral histology. Okay, so from 2017 till 2024, from oral histology, somewhere between 5 to 8 questions were asked. Okay, that's the maximum weightage of this oral histology subject. Somewhere between 5 to 8 questions were frequently asked. Now, let's look at each question. Okay, so from 2017, we are analyzing each question. Okay, and in the right side, you have the main topics. Okay, so after reading this question, we will assign them to the particular topic in the right side where it belongs to. Okay, and at the end, we will see from which topic more questions were asked. Okay, so that's what we are going to do today. So the first question from the 2017 is primary saliva is isotonic, which is modified to become hypotonic while coming into the oral cavity in which part? Okay, it is mainly modified in the striated ducts. Okay, so it belongs to the salivary gland chapter. Next, the anchoring collagen is nothing but it is a type 7 collagen. Okay, so it belongs to the oral mucous membrane. And the blood vessels looped around enamel organ is seen in. Okay, it is seen in the cap stage. Okay, so it belongs to the development and growth of teeth chapter. Next, the first hard tissue forms in 12 to 16 weeks in utero. Okay, so the when can we see the development of the first hard tissue? The first hard tissue to form is the dentine. Okay, usually some people think like since enamel is the outer covering, enamel is the first hard tissue that is formed in the while the development of tooth, but no, enamel is not the first hard tissue to be formed. The first hard tissue to be formed is dentine. Okay, so you should identify which is the first hard tissue formed, and then you should select the correct option from it. Okay, so by around 12 to 16 weeks in utero, you can see development of the dentine. So it is again a development question. Okay, next question. Acantholysis is seen in which layer of oral mucosa? Again, oral mucous membrane question. So it is usually seen in the stratum spinosum. Okay, next question. Which of the following is not a true statement? Okay, the inner layer of periosteum is highly vascular and innervated. The endosteal layer is fibrous. The distance between CEJ and the crustal bone remains constant. And the faciolingual and mesiodistal shape of the interdental papillae depends on formation of tooth and eruption status. Okay. When such questions are asked, you must be really careful. Okay. That is why I highlighted the note here. Okay, which of these is not a true statement means which of these is the wrong statement here. Okay, you should be very careful when they use words like not a true statement or all of the following except, like that. When such words are used, you should be really careful. Okay, read the question very carefully and then choose the answers. Okay, so here option B, the endosteal layer is fibrous. Okay, it belongs to the bone chapter. Okay, next, the taste sensation in tongue is due to which of the following membrane-bound proteins? Okay, it is mainly due to the presence of the gustin. Okay, it is again a oral mucous membrane question. Okay. Now, next, the atypical dentine is formed due to defect in. Okay, the atypical dentine is formed due to defect in histodifferentiation. Okay, belongs to a dentine chapter. Next, the stria of Retzius represents externally on the surface of tooth as it represents externally as pericymata. Belongs to the enamel chapter. Next question, the histopathological staining of melanocytes is done with. Okay, the staining method of melanocytes. Okay, it is again comes under the oral mucous membrane. The intimate lining B cells of the synovial membrane of TMJ are. Okay, the intimate lining B cells of the synovial membrane of TMJ are nothing but they are the macrophages. Okay, belongs to the TMJ. Now, next question, how does the pre-eruptive movement of the tooth occur? Okay, so the pre-eruptive movement of tooth is usually broadly eccentric. So we can put it under the eruption chapter here. Then next, the connective tissue structure that guides eruption of the permanent teeth is, okay, this is gubernacular cord. Okay, the gubernacular cord is like a frequently repeated question. You can either get straightforward questions like this or they can give you an image, okay, and they will ask you to identify the gubernacular canal from it. Okay, so gubernacular cord, very important. Keep reading it. Okay, so this comes under the again eruption. Next, the maximum number of fibers present in PDL is periodontal ligament is these are the oblique fibers. Okay, so we can put it under the PDL chapter here. The matrix vesicles are derived from, okay, the matrix vesicles are mainly derived from the osteoblast. Okay, this osteoblast have a little matrix membrane bound matrix vesicles which will secrete the proteins that are essential for the hard tissue formation. Next, the which PDL fibers prevent extrusion of the teeth? Okay, it is mainly the alveolar crust group. Okay, again, the PDL chapter. The development of pulp begins in. Okay, the development of pulp usually begins in 8th week of intrauterine life. Okay, the development question is very important. Other than the bell cap and the birth stage, you must also know about the development time for the enamel, development time for dentine, development time for the pulp, development time for the PDL, cementum, and the salivary glands, bone also. Okay, so you must be aware about that. You should know the weeks of in utero weeks, which where the development is happening. Okay. Next, the stratum intermedium contains which of the following enzyme? Okay, this is a frequently repeated question only. The stratum intermedium contains an enzyme which is called as the alkaline phosphatase. Okay, this alkaline phosphatase is the main component for the hard tissue formation. Okay, since stratum intermedium is mainly acts as a nutrition for the inner enamel epithelium and the amyloblast and the future amyloblast. So mainly it contains the enzyme called the alkaline phosphatase. Okay, so it is option C which comes under the development chapter. Okay. Next, which of the following is the important buffering agent in saliva? Okay, important buffering agent is nothing but it is the carbonic acid and the bicarbonate buffer. Okay, you can put it under the salivary glands. Next, the organic lining covering the dentinal tubules. Okay, so there is an organic lining that covers the dentinal tubules, which is nothing, it is the lamina limitans. Okay, so it belongs to the dentine chapter. Next, which of the following does occur very rarely about the cemento-enamel junction? Okay, the cemento-enamel junction, what is like rarely seen? Okay, the overlapping of the dentine and the cementum. Again, let's put it under the cementum. The efferent nerve fibers found in the dental pulp are, they are nothing but they are the sympathetic postganglion fibers. Okay, we can put it under the pulp chapter here. Next, according to a ligament traction theory, cells responsible for eruption are, okay, the cells responsible for eruption according to a ligament traction theory is mainly the fibroblast. Okay, so let's put it to the eruption, tooth eruption. Okay. Next question, we have come to the 2020. The component responsible for high intrapulpal pressure in pulp is, okay, so what is the component or that is responsible for the high intrapulpal pressure in pulp? Okay, it is mainly the glycosaminoglycans. Okay, we're putting it under the topic of pulp. Outline bodies are present in which layer of the epithelium? Okay, outline bodies are mainly seen in the stratum spinosum. We'll put it under the mucous membrane. Myoepithelial cells are seen in secretory and intercalated duct. Again, salivary glands. Then the capacity of maxillary sinus. Capacity of maxillary sinus is around 20 ml. Okay, we'll put it under the maxillary sinus topic. Okay, so the next again from 2021 question, the maxillary sinus drains from. Okay, the maxillary sinus usually drains from the posterior 1/3 of the hiatus semilunaris. Again, maxillary sinus topic. Next is as the osteoclasts move through bone, the leading edge of resorption is known as. Okay, so the leading edge of resorption is known as the cutting cone. Okay, so we will put it under the this osteoclast mainly involved in the shedding of process. So we'll put it under the shedding here. The saliva, which is formed in the salivary gland when passes from salivary acini to the duct orifices, undergoes ion exchanges, and as a result, usually the saliva becomes hypotonic in nature. Okay, so we are again putting it under the salivary glands topic here. Most common collagen present in dentin. Okay, so the most common collagen present in dentin is type 1. So we are putting it under the dentin topic here. Next question, the temporomandibular joint development takes place at, like I had already told you, all the structures that are seen here. Okay, you can look at the chapters here and identify, okay, the enamel, dentin, pulp, cementum, PDL, TMJ, salivary glands, and everything. All the development, when the development is taking place, you must be really thorough regarding that. Okay, TMJ development usually takes by 12th week in utero. So we'll put it under the TMJ topic here. True about totipotent cells. Okay. This is a little bit high level topic. The totipotency means it is a type of stem cells. Okay. So stem cells based on their potency. Okay. Potency means the ability to differentiate or ability to divide. They are divided into 3 types. You have the totipotent stem cells, then you have the pluripotent stem cells, and then you have the multipotent stem cells. Okay, so this is like these totipotent stem cells, they are like a newborn baby. Okay, like or you can call it like a student studying in the LKG. Okay, a student studying in LKG has many ambitions, like it can become a doctor, or the student can become an engineer, or student can become an IAS officer. Like the that he or she can become anything she wants. Okay, anything that she dreams of. Okay, so totipotent cells are something like that only. These cells can become anything they want. Okay, so they can go and be an ectodermal cell, or they can be an endodermal cell, or they can be a mesodermal cells. Okay, but when you like learn more and when you advance in age, what happens is that you can only become like a specific type. Okay, like when the age advances, what you can do is like the cell will be more specialized in nature. Okay, the cell will be more specialized means now it can only form like blood cells, or now it can only form like muscle cells, or now it can only form like bone cells. Okay, like that it gets specialized when it ages. Okay, so such type is called as the pluripotent and the multipotency of the cells. Okay, so here they are asking what is true about this totipotent cells. Okay, so these totipotent cells, they can differentiate into 3 germ layers and construct the complete organisms. Okay, so that is a type of stem cell. These totipotent cells are nothing but they are a type of stem cells. Okay, so this is mainly seen in the oral mucous membrane chapter, so we are putting it under there. Then identify the marked structure here. Okay, so till now we have seen like not much image based question, but this is one of the image based questions that can be asked in the dental histology. So the marked structure here is nothing but they are the serous gemmules. Okay, serous gemmules, most of them say that they are not true structures, they are artifacts. Okay, because when under the paraffin embedded section only they were able to see the serous gemmules. The same section, when they take it from the oral cavity and they had done a frozen section. Okay, when they had done a frozen section means they just freeze the tissue and look it under the microscope. So when they did that, they were not able to see the serous gemmules. Okay, so they are now suggesting that the serous gemmules are nothing but they are like a artifact. Okay, it is an artifact that is seen in the paraffin embedded sections. Okay, so this serous gemmules means there will be mainly seen in the mixed glands. Okay, so if this is a mucus acini, then there will be a half crescent shaped structure that will be covering this mucus acini like this. Okay, so these half crescent shaped structure that covers this mucus acini, they are called as this serous gemmules. Now, next question. Plexus of Rashko is found in. Okay, this is from the pulp. The plexus of Rashko is found in cell-free zone of wheat. Okay, so you must be thorough regarding the 4 zones of pulp and the what are the fibers or what are the cells that are seen there also. Okay. Sometimes what they can do is they can give an image of the pulp, of the histology of the pulp, and he will ask you to identify the particular zone. Or the other type they can ask is like they can give the order of the zones of the pulp, like the cell-free zone, cell-rich zone, pulp proper, or dendroblastic zone, and they will ask you to arrange it in the correct order. Okay, so such type of questions can also be asked in the oral histology. Okay, next is antibacterial action of saliva is attributed to all except aminopeptidase. Again, salivary gland question. Supernumerary tooth is due to defect in which stage? Okay, it is due to mainly the defect in initiation. Okay, again a development question. The transient structure determining the shape of the tooth. Okay, it is nothing but it is the enamel note. Okay, enamel note is very important. Lots of researches have been happening now regarding the importance of this enamel note. These transient structures are nothing but they are usually seen in the cap stage of the tooth development. Okay, in this cap stage you will be able to see some structures that will be seen only for a particular time. Okay, only till the cap stage is present, these structures will be there. Then when it progresses to the bell stage, these structures will be vanished. Okay, so such structures are called as the transient structure. So one such transient structure is enamel node, and it is extremely important in the cusp morphogenesis and all. It is mainly like a designer of the tooth. Okay, so it mainly helps in the cusp morphogenesis also. So we can put it under the development topic. Next, the type of collagen that is present in Wonkov fibers. Okay, so Wonkov fibers present in the dentine. Okay, the type of collagen is mainly type 3. Okay, so these type 3 collagen in Wonkov fibers are very important because they are argyrophilic in nature. Okay, argyrophilic means they have the affinity to stain with silver. Okay, so such fibers are called as the argyrophilic fibers. So this Wonkov fibers, argyrophilic fibers, and they are the type 3 collagen fibers. We can put it under the dentine topic. Which of the following contributes to the tensile strength of the bone? Okay, mainly the collagen is on the one contributing to the tensile strength of the bone. Comes under the bone chapter here. Gene involved in odontoblastic differentiation. Okay, these genes are a little bit tough to study because like in the development, if they want to make it a little bit extra hard, then only they will ask about these genes in the odontoblastic differentiation and all. Usually we can put it under the development chapter here. Dentine that is freshly laid over the pulp piece, it is called a predentine. Okay, again a dentine question. Now, amyloblasts originate from ectoderm only. We can put it under the enamel chapter here. Sclerotic dentine under reflected light appear as. Okay, so how does the sclerotic dentine appear under the reflected light? Okay, it appears dark only. We can put it under the dentine topic here. The concentric dark bands or lines present in the enamel as given in the image are. Okay, another image-based questions from the enamel. So what are these concentric lines called? Okay, they are nothing but they are the striae of Retzius. Okay, we can put it under the enamel topic here. The calcified lining of dentinal tubule is. Okay, this is a repeated question. We have seen that we had already answered this lamina limitans questions. Okay, this is a frequently asked question. So the calcified lining of dentinal tubule is nothing but it is the lamina limitans. Okay, we can put another dentine topic here. Now the movement of keratinocytes from the basal layer to the surface is called. Okay, it is nothing but it is a maturation. Okay, we are putting it under the oral mucous membrane topic here. Okay, so in the keratinized epithelium, what you can see that the basal cells, they will start proliferating, and this basal cell population is of two types. Okay, one cell will be there in the basal cell, keep on proliferating. Another cell, what it does is that it will go to the next layer. Okay, so from the basal, it will go to the spinosum layer, then it will go to the granulosum layer, then it will go to the corneal layer. Like that, it will go up and up and up. Okay, so once from the basal layer, it goes up to the corneal layer. That is called as the maturation process. Okay. So when this maturation process is like very high, okay, then only we'll get some pathologies like that. Okay. So besides contraction of the cell, what other functions does the myoepithelial cell perform? Okay, so this myoepithelial cell, they are the basket cells, or they are also called as the cradle cells. Okay, so if this is the acini, this myoepithelial cells will be like this. Okay, just like the half moon shaped cradle, or you can see like a basket. Okay, covering the acini. So what this myoepithelial cells does is that, like the name suggests, it has muscle property and the epithelial property. Okay, that's why it's called myoepithelial cell. Both, it has both the muscle property and the epithelial property. So what it does is that it will just contract, it will squeeze. It is mainly the main function of the saliva, this myoepithelial cells, is that it will squeeze the acini so that more there will be secretion of the saliva into the duct. Okay, so other than that, what this myoepithelial cell does is that it is also an inhibitor of the matrix metalloproteinases enzyme. Okay, so we can put this question under the salivary gland topic here. Next, the excretory duct of submandibular gland. Okay, it is nothing but it is the Wharton's duct. Okay, so again, salivary gland question. Next, we are coming to the recent paper, that is a MDS 2024 paper. Okay, so the stratum germinativum comprises. Okay, this usually I think a lot of students have answered this as stratum basale. Okay, the stratum germinativum is another name for stratum basale, and they had answered that. But no, you should be very careful with the all the options, because in the option D, they have given that this stratum germinativum means stratum basale and the parabasal spinous layer. Okay, that is the correct option. Okay, the stratum basale's another name is not stratum germinativum. Stratum basale together with the parabasal spinous layer. Okay, if this is the basal layer, then the layer that is just above this basal layer, okay, that is the parabasal spinous layer. Okay, so both these layers will have high mitotic activity. Okay, they will be constantly dividing and they will have high mitotic activity. So both these layers, they are together called as this stratum germinativum. Okay, next question. Which among the following is not a cell-to-cell junction? Okay, again, you must be very careful reading this question. Not a cell-to-cell junction, or like except, all of the following except. When such questions came, you should be really careful while reading the question and the options also. So not a cell-to-cell junction is hemidesmosomes, because these hemidesmosomes are nothing but that is a cell-to-basement membrane junction. Okay, cell-to-cell junction are the desmosomes. Cell to basement membrane junction are the hemidesmosomes. Next, the ability of the cementum to repair itself to re-establish the root outline is known as, okay, it is known as the anatomical repair. Okay, we'll put it under the cementum topic here. Which of the following is a major enzyme of saliva? We all know that it is the parotid amylase. Okay, again, salivary gland question. The interdependency of osteoblast and osteoclast in bone remodeling is called as, it is known as coupling. Okay, we'll put it under the bone chapter here. Now, which of the following immune cell is present in the oral mucous membrane also acts as an immune cell for the skin? Okay, it is nothing but it is the Langerhans cells. Okay, you can include it under the oral mucous membrane topic here. Now, the floor of the mouth has. Okay, this question was a little bit confusing for the students. The floor of the mouth is non-keratinized mucosa, mainly due to origin. Okay, now if you look at the option D, that can become a little bit confusing. Like why it is it big or non-keratinized? Because of the ability of the tongue to remove bolus from reaching the floor. Okay, no, but actually the floor of the mouth has this non-keratinized mucosa because of its origin. Okay, so we can again put it under the oral mucous membrane topic here. Okay, so if you see all the questions, most of the questions were asked from this oral mucous membrane chapter. Okay, so majority of the questions were asked from this oral mucous membrane questions, and then the salivary gland topic. Okay, and then you have the bone TMJ and the maxillary sinus. Among this bone TMJ and maxillary sinus, majority of the questions were asked from the bone topic. Okay, and then the next common topic is the dentine. Okay, so these are the four most topics that you should focus on while studying. Okay, so first is the oral mucous membrane, then the salivary glands, then the bone TMJ and the maxillary sinus, and the denti. Okay. Then the development and growth of teeth also. Okay. So I'm not saying that the other topics are very unimportant. You should just brush through the other topics also, but focus. Okay, focus on these four topics. Okay, because if you look at the last 7 to 8 years of question papers, most questions were frequently asked from these four topics only. Now let's see from these four topics which area you should mainly focus on. Okay, so in the oral mucous membrane, definitely you should look at the types of the oral mucosa. Okay, based upon the location, they are classified into like mm, musculatory mucosa, lining mucosa, and the specialized mucosa. All those divisions are given, and based upon the histological feature, they had classified into keratinized and the non-keratinized epithelium. Okay, in the keratinized epithelium, the four layers, and in the four layers, you should be also focusing on the ultrastructural findings of each layer. Okay, ultrastructural findings means all the cell junctions and the bodies that are present inside the cell, like the outline bodies or the keratohyalin granules and all tonofilaments. Okay, you should be aware of all that also. Okay, in the keratinized mucosa, and the staining characteristics also, if you can. Okay, and next is the non-keratinized epithelium. In the non-keratinized epithelium, again the three layers, histology, and the main uh, ultrastructural findings in the non-keratinized mucosa also. And next, you should also study about the non-keratinocytes, or okay, the dendritic cells, the Merkel cells. Merkel cells is a frequently repeated topic. Merkel cells and the Langerhans cells and the dendritic cells, all those things you should be, lymphocytes and all those things you should be reading it thoroughly. Then next is the specialized mucosa, the tongue mucosa. Okay, the papillae of the tongue. The papillae of the tongue, not just the papillae, but the histology of the papillae. Okay, sometimes they can give just a histological picture of the papillae and ask you to identify the papillae. Okay, so histology of the papillae also you must be thorough. Okay, next is the bone TMJ and the maxillary sinus. The bone, the types of bone, the lamellar bone, the woven bone, the uh, the cancellous bone and the cortical bone, all those you should read. Also the histology, the osteon, the Haversian canal, the Walkman canals, and the development, and what else? The mainly that about the bone and the lines. Okay, the structural lines that are seen in the bone, the incremental lines, all that you should read definitely. Then the TMJ again, the development time and the parts of the TMJ and the articular disc and the synovial membrane and the histology also. Okay, mainly in dental histology, read about the histological part also. Okay, maxillary sinus, the lining sinus histology, the capacity, and the draining areas. Okay, that's all enough about the maxillary sinus. Okay, in the salivary glands, another important topic, the types of salivary glands. Okay, major and the minor, and in the major and minor salivary glands, the duct. Okay, remember the duct names of each salivary gland. Okay, the minor salivary gland, and also also some like specialized salivary glands like the von Ebner's gland or the Blandin and Nunn. Okay, like all those terms you should be really focusing on. Then the histological types, the mucus, the serous, and the mixed type, the serous demilunes, and their histological appearance. Okay, you should when you see the histological picture, you should identify whether it is a mucus gland or a serous gland. Okay, you should at least I try to identify that. Okay, mucus glands usually it will be like a foamy pinkish in color. Okay, and the nucleus of the mucus acini, if you see, they will be like pushed towards the periphery. Okay, the gland will be like fully covered and clouded, and this nucleus alone will be pushed towards the periphery. Okay, but in parotid, the serous glands, if you see, the nucleus will be at the center, and the nucleus will be very prominent. Okay, so that's the main difference between the mucus and the serous types, and the functions and the constituents of the saliva. Okay, in the constituents, you should be aware about what function each constituent does, like which one are the antibacterials and which one are the digestive enzymes. You should be aware of that also. Then next is the dentine. Okay, dentine, the types: circumpulpal, mantle, the tertiary dentine, secondary dentine, primary dentine, pre-dentine, the interglobular dentine, then the all those terms. Okay, the dentine is like very important. All those terms you should not get confused, and you must be like really thorough with that. Then the coming to the mineralization, odontoclastic differentiation, not much, but in amylogenesis only they will ask you about the stages of the amylogenesis. Dentine here mainly about the dentine hypersensitivity and the collagen fibers. Okay, this von Koch fibers, which are the collagen fibers, and the argyrophilic fibers. All those you should be really learning. Okay, so that's all about the dentine. So these are the main important four topics, and I had already told you like under these topics what you should really focus on. Okay. Now let's see the type of questions that were asked in the oral history, that can be asked in the oral histology. Okay. So comparatively, when compared to dental anatomy, the image-based questions were a little bit less in the oral histology, but you can definitely expect more image-based and more application-based questions in the future examination. So if you look at the development, you can see that sometimes this the stage of tooth formation like this. Okay, this is the advanced bell stage. So such stage of tooth formation can be given, image can be given, and they will be asked you to identify which from which stage of tooth development is this. Okay, so you should be really looking into the histopathological pictures of the each stage, how it looks in the bell stage, how it looks in the cap stage, how it looks in the bud stage. You must be thorough with that, and then you should answer it. Okay, next is in the cap stage there is some special structures that are called as the transient structures. I had already mentioned about this transient structures. Okay, so these transient structures, sometimes they can give the picture of these transient structures like this and ask you identify that also. Okay, so that, and next one is the amylogenesis. Like I had already mentioned, amylogenesis is important. The stages of amylogenesis are important. Sometimes they can give a picture like this and ask you to identify which stage is this. Okay, when they ask that which stage is this, you must be able to answer that also. Okay, next is they can give a ground section like this. Okay, this is the ground section of enamel, where they were mentioning ask you to identify about the gnarled enamel. Okay, so gnarled enamel, the dead tracks, and all those things you should be able to identify. Okay, then these structures also, like the enamel spindle, enamel lamellae, enamel tuft, all those you should be thorough with in the enamel. And again the these things also, prenatal enamel, postnatal enamel, and the line between them. What is the line called? Okay, that is the neonatal line. Okay, and then about the cementicles. Okay, so cementicles. Okay, these are the small structures. These are called as the cementicles, and denticles also. Okay, so which type of denticle is this? Is this embedded one, or is this the free one or attached one? Like that also they can ask. The type of the cementrical also they can ask. So what you can do to study these image based questions are, like, take a standard textbook. Okay, uh, for oral histology I will prefer you to take either the Orban's oral histology or the 10 Kate's oral histology. Okay, so these are the two standard books for the oral histology, either the Orban's or the 10 Kate's. Okay, then while doing revision or while studying, just go through the pages of this oral histology textbooks. Okay, just go through the pages, and whenever you see an image, just look at the image and read the subheading under it. Okay, do it for like 2 or 3 times. Okay, now after 2 to 3 times, what you can do is like again go through this textbook, and when you see an image, like hide the subheading under it. Okay, hide the subheading under it and try to remember what this image is about. Okay, so this is one way by which you can tackle the image based questions. Okay, not just for oral histology, any subject, this is one of the ways to tackle the image based questions. Okay, so usually images stay better in our mind than the words also, so try doing this. Okay, now since I had already told you this oral histology is a basic subject and it's like a lab subject. Okay, so uh, uh, how can an application based or clinical based questions can be asked from these topics? This is a typical example of that. So here they are giving like a child suffers from a febrile illness. Okay, during the illness, the tooth development temporarily slows down. Okay, the development resumes normally soon after. This change in development of the tooth structure is most likely to be apparent in which of the following histological features of dentin. Okay, so this histological feature of this change. Okay, so usually what will happen is that in the this hard tissue formation, they will be happening in layers. Okay, so these layers, one layer will be depositing, and then they will. It's like a period of rust and period of activeness. Okay, so once they will deposit the layer, then they will take rest. Okay, the cells, they will take rest and then they will again produce this and then they will again take rest. So that's why we are, when doing ground section or when doing decalcification, we are mainly the ground section, we are able to see lots of lines in these hard tissues. Okay, so these lines indicates that these cells, they are not continuously forming these hard tissues. They are in a period of rest and a period of activity. Okay, so when some external agents are affecting it, like the illness that is mentioned here, what happens is that there will be like a period of rust or like the hard tissue formation will not be like the normal. Okay, so what happens is that there will be changes in the particular structure of the hard tissue. Okay, so in dentin, in which layer or in which these changes are seen, that's what they are asking. So it's mainly seen in the lines of von Ebner. Okay, the incremental lines of the dentin, lines of von Ebner, mainly the changes are happening. Okay, so expect such type of questions also, such application or clinical based questions also for the oral histology. Okay, so these are the total type of questions or the pattern of questions that can be asked from oral histology. And I had already mentioned the high yield areas that you should focus on and the topics under each high yield area also. Now, that's all for today. If you have any doubt, you can comment under this video. We'll definitely answer for that. And also, we'll be coming up with more such live sessions for each subject. Okay, so stay tuned to Edentu YouTube channel. Okay, so that's all for today. All the best for your preparation, doctors. Do well. Good night.